Hi class, Dr. Jim here. In this lecture, we're going to discuss a little bit about transport. Now, in the previous lecture, and hopefully you've watched that already, I discussed the structure of the membrane. Now, today what we're going to look at is actually the function of the membrane and how it is semi-permeable. So in the last lecture, I mentioned that it has a lot to do with the molecule. If it's small and non-charged, it can easily pass through, and that's called diffusion. And we're going to look at some examples of that today. If they're large or they have a charge, it has to get through by some other mechanism. It doesn't work by diffusion. It either works by what is known as facilitated diffusion or by active transport. And active transport requires energy, and that's why it's active. And we're going to look at all these different examples today using the membrane and using these various forms of transport throughout. So let's take a look at these different things. So our objective is how do molecules move across the membrane? And I've already discussed, and if you watched the previous lecture, it all depends on the type of molecule. If it's small and no charge, or small or doesn't have a charge, it can easily pass through. If it does have a charge, or if it's very large, then it has to use some other mechanism to get across the membrane. One way to help regulate that. Then we're going to look at the different types of passive diffusion. So passive, the word passive here is very important. Passive means that you do not expel any energy. No ATP is used during this process. It means that everything can freely cross the membrane and the cell does not have to expend any energy to do it. The other type is active, and active diffusion or active transport means that you require energy. So always associate active with energy burning using ATP, and that's what's happening in this situation. So let's take a look. So on here I have a nice video for all the different types of transport. It's about a 5-minute video, and again I recommend watching this after. It just kind of gives you an overview of all the different types of transport that's associated. We're going to talk about passive transport when no energy is used, and then active transport. And this just kind of gives you a nice video montage of what everything is. And so highly recommend just watching it, taking it in, and kind of refreshing everything that I talk about here in the lecture. So take a look at this video once you get done with this lecture. So the first type of movement, and again, if the molecule is small or doesn't have a charge, it can easily move across that membrane. And that is called diffusion. And so diffusion is the movement of molecules. And typically what we always associate in biology is that in biology everything wants to be equal. And so if you have something that is of high concentration, it's going to move to an area of low concentration so it can equalize the amount out. So you want to have the same here as the same on this side of the membrane. So you have equal amounts. So if you have a high concentration on this side, everything is going to cross the membrane like this to go to the low side, okay, so that you get equal amounts on both sides. And that's how biology works. And so you can do this experiment at home. If you take a packet of Kool-Aid and put it into a pitcher of water, what happens? And so all you do is you pour that Kool-Aid into that pitcher, and you see that it starts very concentrated in a little spot. If you let it sit there without mixing it up, eventually that Kool-Aid will come throughout. It will take a while, but it does. And so that's the equal part of it, so that that high concentration will go to low concentration. And that dispersal is just known as diffusion. So that's the movement of molecules from high to low concentration. So get that in your mind there. Diffusion is the movement of molecules from high to low concentration. Very easy passive transport. Okay? We use this concept in our lungs. Now our lungs use this process to exchange oxygen into our blood and then taking carbon dioxide waste back out, and that's what we exhale. So the process, think about this. As you take a breath in, like so, you're bringing in oxygen. Oxygen goes into the lungs, okay? It travels into the lungs, and then what happens is once oxygen comes in from the lungs, it goes into the blood vessel, because you think about what's in the blood vessel before it gets oxygen is a lot of carbon dioxide. It's very non-oxygenated blood, okay? So oxygen is going to go in by the process of diffusion, and likewise at the same time as you put oxygen in, carbon dioxide is going to enter into the alveoli of the lungs and then work its way out. So as soon as you take a breath in, like this, you exhale and you breathe out carbon dioxide. And so it's that process of diffusion that allows that to take place. And so it shows you really on a human scale what diffusion really does in our bodies. Okay? Another type of movement is the movement of water. So we call diffusion of water by a special name. We call that osmosis. Now, some of you may have had parents saying, how are you going to learn this material? By learning it through osmosis. But really what they mean is maybe by diffusion, because you sleep on the book and maybe it'll somehow enter into your brain. But osmosis is the special type of diffusion known as the movement of water across the membrane. And again, it's going to work in a situation based on concentration. Now, you kind of have to think about it in the opposite direction than I just talked about. Diffusion is from high concentration to low concentration. Water kind of works in the opposite principle in saying that it wants to dilute the high concentration and get rid of the low concentration. Okay, so water is going to move in the area where you have high concentration. So let's look at this example that I have on the slide here. Let's say you have a lot of sugar on one side and not a lot on the other. And you separated this out by a membrane that can't move. And you're going to do this in the lab this week. And I don't want to give away all the fun that you're going to do in the lab, but you're going to actually do an experiment like this where you have a semi-permeable membrane called a dialysis tubing. And it allows some things in and out and doesn't allow other things in or out. Okay, and it's all based on size. So if you're small, you can get through. If you're too large, you can't. And so that's something to think about when you're doing the lab this week. Okay, and so that membrane allows water to pass through, but it doesn't allow the sugar. The sugar is too big to get through. So in this situation, the concentration of sugar can't go to the lower concentration. It can't diffuse across that membrane. So what happens instead is water comes in to dilute. And so water is always going to try and dilute the higher concentration. And so this is really important when we start talking about these big words, hypertonic, hypotonic, and isotonic in just a minute. So water is always going to move in the concentration where there's more solute. Solute is what gets dissolved. And so water is going to move in that direction. And so you can see in the example is that the water concentration on this side goes down and increases on this side because it wants to dilute the sugar. And so that's really important. So that's the idea why we get this hypertonic, isotonic, or isotonic and hypotonic solutions. Okay, and osmosis is the movement of water across the membrane. So that's the definition of it. So let's look at these different examples. I just talked about hypertonic, isotonic, and hypotonic, and you might wonder, what does this really mean? Well, hyper means that you have, so let's describe this as a beaker and putting a cell into that beaker. So in that beaker, you have a beaker of water that has lots of salt. So think of hyper like a hyperactive kid, lots of sugar. Well, in this situation, you have lots of salt in the water. Now you put in that cell, and what's going to happen? So if we think back to that example we just showed where you had more sugar on the one side and all the water rushed in or ran to that other side, the same thing happens in the cell. If I have more salt on the outside of the cell, water is going to rush out of the cell and try and dilute that salt on the outside. So what happens to that cell is it begins to shrink because it loses that water. And that's known as crenation or dehydration. And so this is what happens to you when you start getting, you stop drinking water and you get really warm, you get really hot, and you get dehydrated. This is what happens to your cells. They shrink. And if you don't get enough water, you start to feel agony because your cells are kind of screaming to you, hey, get us some water. And that's one of the reasons why you never drink seawater. Because if you were stranded on a boat and the only water you had was the ocean water, it'd be horrible for you because all you're doing is putting more salt into your body. What that salt does to you is that it forces more water out of your cells and actually dehydrates you faster. So you would actually die faster by drinking the seawater than if you didn't have any water at all. So that's one rule of thumb, never do that. Now, the other situation is called hypotonic, and hypotonic is when you have more salt on the inside of the cell than you do in the beaker. So let's say you put the cell into normal tap water that we get out of the faucet or the bubbler. Okay, you put the cell in there, And what happens now is, again, more salt on the inside of the cell than on the outside. And so what's going to happen to the water? The water is going to rush in. And that keeps filling into that cell until the cell can't take it anymore. And eventually what it does is goes kaplooey and blows up the cell. Okay? The cell blows up and now the cell is dead. This is what happens if you gave someone an IV of water. Okay? You're always told give someone an IV of a 0.9% saline, and the reason for that is our cells are about 0.9% saline. If you gave them pure water out of the faucet and gave it as an IV, you would lyse all their blood cells, they would die, and you would probably lose your job. So never do that for your patients. So there you go. There's your rule number 2. Don't drink seawater. Don't give a patient regular water as an IV. Okay? And the third situation is called isotonic, and isotonic actually means equal. Okay, it means equal concentrations of salt. And in this situation, water can move back and forth very freely. It doesn't just stand there, it actually moves back and forth, but you don't have these crazy changes or osmotic changes that cause the cell to either shrink or to blow up. And so that's the key. Now this also happens in plant cells. And in plant cells we have a few other definitions, and so make sure that you know the different types of things that happens depending on the cells that you come in contact, because we do on the test ask you about the difference between plant and animal cells. In animal cells, when you get dehydrated, that's called crenation. When you burst, that's called cell lysis. And in plant cells, you have a couple of different things. So when the cell in plant cells, or the water rushes out, so in this situation, again, dehydration, what happens is that you have the wilting of the plant, also known as plasmolysis, where the plasma, or the plasma inside the cell, or the cytoplasm, actually starts to pull away from the cell wall, and you can see how it shrinks down. And this is what happens to your garden plants. When it doesn't get enough water, it wilts because it doesn't have enough pressure to keep it upright. And so that's what happens when it dries out. Isotonic, the plant is nice and healthy, the water moves back and forth, there's no change. Now, a big difference between plant and animal cells is when you put in a hypotonic situation. When you put in a situation, that cell wall actually prevents the plant cell from bursting, and actually the plant really likes that situation. That's why you give your plant normal tap water, because what happens is that you make the plant very strong and tall. It's called turgid pressure or turgid because it becomes nice and strong. It's that osmotic pressure that pushes against the cell wall that keeps it solid, and that's really important. So you want to make sure that the cell remains nice and solid in that situation, and so that's called turgid pressure, and that's what the plant wants, and that's what happens. It doesn't blow up in the hypotonic, it actually gets stronger and more structured. Okay, and so that's hypertonic, isotonic, and hypotonic. So make sure you know what happens in each one of those situations. Okay, the last type of passive diffusion is called facilitated diffusion, and this is when you have a large molecule that has a high concentration on the outside and a low concentration on the inside, or vice versa, and all you want to do is pull those things across. Take it as like sugar being on the outside. You have lots of sugar on the outside and it wants to get on the inside. You need a helper to get it in because it's just too big to get across, and so it holds out its hand. And this is facilitated diffusion. It's movement from high to low, and it needs a carrier but no energy. This is like you going down the river and floating with the current. There's no energy required. You just relax. You're going from an area of high concentration to low, and so that's the big thing. The next type of diffusion is actually now when you exalt energy, and when you use energy, that becomes active, and active transport is when you use energy to move things across. The big difference not only is using energy, but now you're going from a concentration of low concentration to high, so you're actually doing the opposite of what you normally do. And the reason for this is maybe you'd like to hoard things inside your cell. Let's say you have a high concentration of sugar in your cell and you don't want to lose any of that sugar over time. But in normal biology, that high concentration wants to move it to low concentration. Okay, that's just normal activity, but you want to keep it in, you want to hold it in, and in order to do that, you have to use energy to keep all that sugar in because you don't want to lose it to the outside. And so when you use energy, you're doing active transport. And so again, it's movement across the membrane. It's when you have a large molecule or a charged molecule, but you want to do it in the opposite situation. You have low concentration on one side, high concentration on another, and you want to keep it in that situation. You want to keep it low on one side and high on the other. So you have to use energy. And I use the example going against the current, because that's what you're doing. You're going against the current in that situation. You're using energy. You struggle. You're swimming. Use that energy to go against the grain. Okay? And so that's important. Now, there are actually 3 types of active transport. So we talked about the simple molecules coming about, but these are when you have large quantities of things that the cell wants to get in. And this is seen really in the eukaryotes, not so much in the prokaryotes, but it does happen. So all these things happen. This is active transport, but this is more like with phagocytosis and things like that. So there's 3 types. There's something called phagocytosis, and this is kind of the food particles. This is the eating. You also have a thing called pinocytosis. This is kind of when you bring in molecules of water in big quantities, and that's kind of like you're drinking. And then you have this thing called receptor-mediated endocytosis. And the only difference between this and phagocytosis is that you're binding to a receptor. Remember the receptor proteins I talked about in the last lecture? You're actually binding things to it and then bringing it inside the cell after binding. And so I'll show you an example of each one of these. So the first one is the phagocytosis. And so what happens in this situation is you got food particles that need to get in. Your cell surrounds it. Your membrane kind of swallows it up like a Pac-Man, and eventually it surrounds it all the way around and then brings it into the cell. And you can kind of see this, where this cell here, this amoeba, is about ready to eat this little paramecium, and so he's going to surround it and then bring it in and eventually eat it. And that's called phagocytosis. The next one is called pinocytosis, and this is where you're drinking. Now, in this situation, you have some kind of solute or water or something else. Again, you bring these molecules in and you cross the membrane. You can see here a number of different vacuoles kind of forming and, again, bringing in whatever's on the outside. And this is like large doses of water that you want to bring in at one time. And so we call this pinocytosis, or known as drinking of the cell. Okay, and then the last one, again, is the receptor-mediated. And what you have are these proteins. We talked about these proteins before. They bind these molecules on the outside. When you bind these in and attach, it sends a signal to the cell, hey, we want to come into the cell. The cell then makes this, again, this invagination occur. It pinches off and then brings these inside the cell. And a lot of times this is utilized in immunology and immune cells to signal something and bring things in. And again, you see these from time and again, and this is called receptor-mediated endocytosis. So all 3 of these things are bringing things in, which is called endocytosis, but they're all different examples depending on what type of molecule you're bringing in. Okay? Now the cell also wants to get rid of stuff, and it gets rid of waste. And by the way it gets rid of waste is by actually called exocytosis. So endo is bringing in, exo is leaving, and so that's the process of leaving. You're bringing waste outside the cell. And so when you've digested the food or you have waste in the cell and you want to get rid of it, you actually bring it to the membrane and then you release it out. Just kind of like, if you ever remember the Star Wars movies and the trash compactor and they release it out into space, it's kind of the same way. Same idea is that it moves to the membrane, and then as soon as it gets to the membrane, it opens up and releases these waste products. And sometimes you can do this also with any secretory proteins or anything else. So like the Golgi will make something, send these vesicles to the membrane, and then it will release it out to other cells or other organisms, depending on what type of molecule or organism you're talking about. Okay, so to summarize, there's 2 types of transport. There's passive and there's active. Passive, again, you're going from high concentration to low concentration. No energy is required. That's the most important thing with passive. No energy required. And there's the 3 types. There's diffusion, in which simple molecules are small enough to get across the membrane. There's osmosis, which is the movement of water. And remember the 3 types: hypertonic, hypotonic, and isotonic. And then facilitated diffusion, when you have large things that have to get across using the protein carrier. You have then active transport, and what makes something active? Energy. And again, you're going across the grain. You're going from low to high, and you need that energy. And in order to do that, you have to expel energy. You're going against the current. Okay? Okay, and that's the big thing. And we talked about this before, the 3 types of endocytosis, getting things across the membrane, bringing them in. So you have the drinking, the eating, and the receptor mediated, and then finally the exocytosis, when you release things. And so those are all different types of ways that we get it. So make sure you remember passive and active. What makes something passive? What makes something active? And remember, in passive transport, the different situations, because we like to ask about hypertonic, hypotonic, and isotonic. So make sure you have that down. If you have any questions, please feel free to email me or ask me in lab. And I'm happy that you're watching these videos, and I'll talk to you soon.